Can somebody confirm when it's on? Where? Good. Practice makes, if not perfect, better. We tried it with Juan Carlos on Friday. There we go. Okay, well, good morning, everyone. It's 11 a.m. in the Eastern US, noon for our speaker today in Argentina, and who knows what time for some of our visitors and participants who are joining us from all over the world. Welcome you all. And before I introduce our speaker today, I'll just go very, very quickly, although it looks like everyone in the audience has been here before. We'll just have our speaker present for 50-55 minutes. And after that, we'll follow with a session of questions and answers. For that, we ask you that you type in the chatting window a question mark. That way you know when you're coming up to ask your question. We will encourage and give priority to our young students and scholars and also the community, the YouTube community, to join us asking questions. Before I introduce the speaker, let me just mention that next week, October 5th, we're going to hear from Clint Pennick from Kennesaw State University in the USA. And he's going to be discussing dominance hierarchy formation and brain changes in a socially flexible ant. So we're up for another very interesting talk next week. Today is a very special speaker who joins us, a very special definitely for me, Dr. Juan Carlos Reboreda. He is from Argentina. He's a biologist by training who completed his doctoral degree at the University of Buenos Aires in Argentina. And then following his PhD, he went on to do postdoctoral stays in the UK, primarily actually England, and splitting his time between Cambridge and Oxford. And for those of us with some years on our shoulders, I'm sure you'll enjoy learning that he was fortunate to spend some time with John Krebs, with Alex Kaselnik, with Nick Davis, who apparently was the one getting him all excited about studying parasitism. Juan Carlos, when he went back to Argentina, he established himself as a researcher there. Today, he is a professor at the University of Buenos Aires. He's also a researcher in the CONICET, which is the National Council for Research. And he's the director of LEICA. LEICA is the name of his lab, which stands for the Lab of Animal Behavior and Ecology, which he founded in 1998. He has had a very productive career, and something when we kind of look at these very extensive CVs and try to take some notes, something that really caught my eye is that he has advised over 20 PhD students over these years, and an equally impressive number of master students. When somebody only lists articles for the last 10 years, that's already telling you something. And I counted about 70 articles published in the last 10 years. So he has continued to be very, very productive. And not only while being a professor and directing the lab, but the last things I want to say about Juan Carlos is that he's continued to be productive while being the dean of the School of Exact and Natural Sciences in the University of Buenos Aires. And just to give you a little bit of scope to what this task must entail, he was sharing with me some numbers of the University of Buenos Aires. The University of Buenos Aires is the largest university in our country with 300,000 students. And the school of which he's the dean has 1,500 PhD students. About between 200 and 200 PhD students complete their degrees per year. And this is a massive amount. I mean, this is a huge institution. That counts for about 10% of all PhDs given in Argentina overall for the whole country. The school has roughly 400 professors, and very much like the research universities in the US and Europe, all of those professors, they're 100% devoted to research and teaching. They're full-time employees of the university. What else I can mention that was interesting for me? This is the university and the school from which I graduated with my first degree. So that's why I was very interested in getting a little bit of background to understand. They're publishing roughly 1,200 articles, peer-reviewed articles in index journals a year. So you can imagine handling a faculty, a school like that. It must take a lot of time. Still, he has managed to remain active. And today he will be, I can share my screen and show you quickly. So much of his research has been centered in the study of interspecific avian brood parasitism. And today he will be presenting to us a talk titled Coevolutionary Arms Race between a specialist avian brood parasite and its host. With that, I would like to invite Juan Carlos to take it from here. Juan Carlos, I am making you now co-host so you can share your screen and take it from here. Thank you so much for joining us.
Okay. Well, thank you very much, Eduardo, and thank you very much, all of you, for participating in this seminar. As Eduardo told you, I'm going to talk about interactions between a specialist brood parasite and its main host. Let's start with, sorry. Yep. For those of you which are not familiar with brood parasitism, it's a form of breeding biology with individuals that exploit the parental care of unrelated individuals. And you have intraspecific and interspecific brood parasitism. I'm going to talk about interspecific brood parasitism. And it has evolved in insects, and insects always are good examples for everything. But you have also brood parasitism in fishes, but perhaps the most well-known examples are in birds. And since Aristotle, it's known that some bird species have this peculiar way to reproduce. And at present, you have brood parasites in about 109, 110 bird species. They are in 27 genera, 5 families, and here you have examples. Perhaps the best known examples are, are the cuckoos, but also you have the Indicatoridae in Africa, the Biduidae in Africa, the black-headed duck, which is the only altricial, the only precocial species, interspecific brood parasite with precocial young, and the cowbirds. And brood parasitism imposes fitness costs on the host reproductive success, and these are main cost. It reduces the clutch size either by removal or puncture of host eggs, also reduce the hatchability of host eggs, the survival of host nestlings, either because brood parasites evict, kill, or just compete for food with host nestmates, reduce the post-fledgling survival of host young, and also it can reduce the survival of the future reproductive success of host parents, and it increases the desertion of the nests which are parasitized, or the depredation of nests which are parasitized. And all these costs selected for the evolution of antiparasitic defenses, and by antiparasitic defenses, I mean traits, either morphological, physiological, or behavioral, that reduce the impact of parasitism. And these host defenses may select for counter defenses in the parasite, and this potentially could generate a coevolutionary arms race. And it's important to emphasize that defenses and counter defenses can act at one or at several stages of the nesting cycle of the host. I mean, the egg stage, the nestling stage, or the fledgling stage. And here in this table are the main defenses and counter defenses. The first line of defenses is aggression towards the parasites, and the counter defense is elusive behavior, resistance to the host attacks, or rapid egg laying. If this line of defense fails, then you have the possibility to reject discriminate and reject either by ejection or necessation, reject the parasite egg. And the counter defense is the mimicry of host eggs. And the third line of defense is to discriminate and reject the chick, the parasite chick. And the counter defense is mimicry of host chicks. And here you have two examples of mimicry of eggs in the common cuckoo. And here in the pictures, you have two different hosts. And the larger eggs is the cuckoo egg, which is mimetic with host eggs. And the example of mimicry of host chicks are in this Vidua brood parasite with his host. And in this case, you have visual and vocal mimicry. And I just want to show you a video. I will show several videos. This video is not with the species I'm going to talk, it's with the other brood parasite common in Argentina, the shiny cowbird. And here you have one nest of the chalk-brow mockingbird. That's the host. And the first bird who entered the nest is the shiny cowbird. And you can see the aggression of the host towards the parasite. This is the first line of defense. You can see now the same from a different angle. There were two eggs in the nest. Now you have 3 eggs, and now 2 eggs. So in this video, you have defenses and counter defenses, rapid egg laying, resistance to host attack, aggression to the parasite, and egg ejection of the parasite egg. So I'm going to talk about brood parasitism in cowbirds, in particular in one species of cowbirds, Molothrus rufaxillaris, from now on the screaming cowbird. And the cowbirds are a monophyletic group, which includes 6 species. And these species differ in the degree of specialization in host use. Molothrus rufaxillaris, the screaming cowbird, uses almost exclusively one main host. At the other extreme, you have the brown and headed cowbird and the shiny cowbird, which have more than 200 different hosts. And this group, this group is about 4 million years old. Sorry. So let me introduce the screaming cowbird. They are dimorphic in body mass. Males are 20% larger than females. They are monomorphic in plumage coloration. And as I mentioned, it uses almost exclusively one host, the baywing. And this specialization includes the plumage and the vocalizations of chicks and young that resemble the plumage and vocalizations of the main host. And here you have the eggs of the host and the parasite. But this is how the host and the parasite looks when they are 6 days of age. This when they are 12, 14 days of age. This is when the parasite is about 40, 50 days of age. And this is how they look when it's adult. So the young parasite start to mold into the adult plumage at the time they become independent from foster parents and join conspecific flocks. How about the host? The host is sexually monomorphic in plumage and in body mass. It's a cavity nester and it's a cooperative breeder. Helpers, they have helpers in approximately 50% of the nest, and helpers primarily are males, which are genetically related with the breeding pair, but you also have unrelated helpers, males and females, but most helpers are males and are genetically related with the breeding pair. Is the main host of the screaming cowbird and the frequency of parasitism is more than 90% of the nests, with an intensity, by intensity I mean number of parasitic eggs per nest, that varies between 3 and 5 eggs. And this host is also parasitized by the shiny cowbird, the generalist brood parasite, and the shiny cowbird have chicks which are non-mimetics with the host. It parasitizes this host at a lower frequency between 15 and 20% and with a lower intensity of parasitism. So we have a system in which we have one host that is parasitized by one specialist brood parasite and one generalist brood parasite. The specialists have vocalizations and plumage that resemble those of the host, and the generalists have differences in plumage and vocalizations. And this is our study system. So which are the costs that screaming cowbird parasitism produce in baywings? Well, there are two main costs. And this video will show you an event of brood parasitism. Here is a female, and as you can see, they start pecking the eggs in the nest. There were 2 host eggs. And as a result of this behavior of pecking and puncturing, it reduces the clutch size of the host, and the impact is proportional to the intensity of parasitism. And you can see now that this is a rare event. This is a second female screaming cowbird. They arrive together. This is not unusual, and both are trying to destroy host eggs. And you can see that they both lay 1 egg. So now have 4 eggs. And this increases the rate of nest abandonment. If the destruction of eggs is important, the host can just desert the nest. But there is no significant effect of parasitism on the hatching success of the host eggs, or in the nestling survival, or in the body mass of host fledglings. So main costs are egg destruction. So I will show you defenses and counter-defenses, first before and during the egg stage, then during the nestling stage, and finally during the fledgling stage. So as I mentioned, the first line of defenses is aggression towards the parasite females, and the way to test if hosts have this antiparasitic defense is to run an experiment in which we present taxidermic models of the parasite and a control species. In this case, we presented taxidermic models of screaming cowbirds, shiny cowbirds, and a control species. Was a within nest design in which we did repeated measures. We present one model for 5 minutes, then an intertrial interval of 15 minutes, the second model, and then the third model, and the order of presentation of models was rotated among nests, and we measure the approaches to less than half a meter from the model, and the frequency of attacks to the model. And the results show that baywings attacked more frequently and spend more time close to a screaming cowbird than to control models. These are the values. This is number of attacks. This is proportion of time near the model. These are the results for screaming, and is quite different from control. And shiny cowbirds have an intermediate response. So baywings recognize parasitic females as threats and direct aggression preferentially to the species that more frequently parasitized the baywing nests. Another defense is what we call unpredictable start of egg laying. This graph shows the length of the pre-laying period in days. Pre-laying is the time since nest lining finished. You can predict when egg laying will occur because the last step in nest building is the lining of the incubation chamber. And normally 1 day or 2 days after nest lining is finished, the bird start laying. In baywings, this pre-laying period is not 1 or 2 days, varies between 1 and 19 days. And we can say it's not statistically unpredictable, but we can say that it's unpredictable to make synchronization of parasitism difficult for the parasite. And as a result of this, in 70% of the nest, there are parasitic events during pre-laying, and all parasitic eggs laid during pre-laying are ejected. And in this way, the host deplete the number of eggs in screaming cowbird populations. 50% of the eggs laid in baywing nest are ejected because they are laid before the host start egg laying. The other antiparasitic defense is egg protection against parasitic attacks. And again, I show you a video. This is the baywing in the nest, and one female screaming cowbird arrived, and you can see that it tried to peck the eggs, but the host is protecting the eggs. And finally, it lay an egg on top of the host. So this behavior doesn't avoid parasitism, but avoid the destruction of eggs. And in more of parasitic events, in 70% of screaming cowbird visits, baywings females were inside the nest. And when they are inside the nest, egg punctures occur at a lower frequency than when they are not inside the nest. So this tight-sitting behavior prevents screaming cowbird females puncturing host eggs, and therefore reduce the cost of parasitism. The other antiparasitic defense is ejection of parasite eggs. As I mentioned, baywings nest they nest in cavities, so light is very low, and because screaming cowbird eggs are relatively similar to host eggs, discrimination of eggs is difficult. So they eject cowbird eggs if they are laid before the host start laying, but once the host start laying, it stops ejecting cowbird eggs, unless the number of parasite eggs is quite large. And these graphs show the probability of ejection to eject the whole clutch as a function of intensity of parasitism. As you can see that if intensity of parasitism is more than 3 or 4 eggs, the host ejects the whole clutch. The host don't recognize parasite eggs, but in some way recognize that this is an extraordinary clutch size. And as a result of that, of course, after ejection of the whole clutch, it starts a new clutch. And it not avoids being parasitized, because in the new clutch is also parasitized, but the intensity in this replacement clutch is lower than the clutch that ejected. So this behavior allows the host to reduce the intensity and the costs of parasitism. And this is a video just showing the way in which the host eject eggs. Most hosts eject eggs with the bill, grasp and eject the eggs, or puncture and eject the eggs. But this host have a bill which is small, so the way they find to eject eggs is using the feet. Well, which are the parasite counter defenses? Well, one behavior that cowbirds have is to prospect first search and find a host nest, and then prospect. So visit regularly the host nest until the host start laying. And at that time, they parasitize the nest. They synchronize parasitism, visiting regularly the nest until the host start egg laying. So the way in which we detected, measure visits of parasites to baywing nest was fitting the parasitic females with VHF coded radio tag. These radio tags emitted in one frequency, just one frequency, and have one ID code, so you can identify up to 50 different individuals, sorry, 500 different individuals. And we put one proximity, one receiver with data loggers connected to an omnidirectional antenna below the baywing nest. And we set the receptor to detect only tags that were within a radius of 20 meters from the antenna. So these data loggers were continuously listening for tags and recorded the date, the time, and the intensity of the signal. And the intensity was inversely proportional to the distance from the antenna. So in this way, we were able to monitor the visits to parasite, so to host nests for long periods of time. In fact, we normally put the data logger 5-10 days before during the pre-laying period, and continue recording data until mid-incubation, so between 15 and 20 days. And this graph showed the results of 5 different females. And you have, in all cases, in the y-axis, the number of visits, and in the x-axis, the day. 0 corresponds to the day the first host egg was laid, and negative values are pre-laying, positive values are laying and incubation. And the white bar show visits to between 10 and 20 meters, and black bars visits to 0 and 10 meters. So we don't know if the parasite was inside or outside the nest. They are just visits to the proximity of the nest. But you can see that you have between a few, 3, 4, up to 20 visits, 15, 20 visits in a single day, but the same female. And that happened several days before the event of parasitism, and the arrows indicate the time at which the parasitic female lay an egg. We have 2 cases of repeat parasitism, which is unusual, because in most cases, the parasitic female lay only 1 egg per nest. You have multiple parasitism because different females lay in the same nest, but every single female lay only 1 egg. And we are relatively sure about this because we extracted maternal DNA from adult females and from the membranes of eggs unincubated, and genotyped the eggs and, well, the samples of DNA. We use 6 polymorphic microsatellites and assign eggs to unique maternal genotypes. And what we found was that in only 2 cases, 2 of 57 nests, we had repeat parasitism. So 1 female that lay more than 1 egg. In all the other cases, we have that females lay only 1 egg per nest. So they apparently avoid repeat parasitism. And this is interesting because they appear to have a mental inventory of host nests. So they once a nest is parasitized, is removed from this kind of library of nests they have, and they avoid the competition of competition between their own chicks. Another peculiarity of this brood parasite is the timing of parasitism. These data were obtained with micro cameras in the nest. What you have in the x-axis is the time relative to sunrise. So this sun indicates the time of sunrise, and the black bars indicate parasitic events, and the white bars, the departures of the roost, the place where screaming cowbirds spend the night. And you can see that the time of parasitism is between 40 and up to 60 minutes before sunrise in almost total darkness. And something which is interesting is that apparently the days they are not parasitized in nests, they stay longer in the roost. They, like humans that stay longer in the bed during the weekends, while the days females are not laying an egg, they stay longer in the roost inside. And this time of parasitism may help the parasite female approach and detect it to the baywing nest. And also, this can reduce the probability of detection when the baywing is not inside the nest. Another peculiarity of this species in particular, there are some theoretical discussion about the mating systems you expect in a brood parasite. You don't expect monogamy or social monogamy, because the main explanation for monogamy in birds is parental care, biparental care, the need of biparental care. So in absence of parental care, what you expect is more promiscuous mating systems. But normally you see male and female, you see pairs of screaming cowbirds. And when you trap, normally you trap one male and one female. So one possibility is that males could help females in locating and prospecting host nests, or in facilitating the access of females by distracting hosts, for example. So we again fit pairs of screaming cowbirds with radio transmitters, the female and the male that were caught together, and we create an array of 4 these data loggers, these receivers. Just to give an idea, the distance between these violet dots is about 1 kilometer. We have an area of approximately 150 hectares. This is the place they used to roost, and the orange dots show active nests during the time we collect the data. And what we did was just to track simultaneously females that were within this area, and then females and males. And then we did a social network analysis to see the association. And in all cases, 9 pairs that were trapped together, they were associated most of the time during all the day. We did this for almost 1 month. The radio transmitters last for 60 days, but we have data for approximately 30 days. They are socially monogamous, they stay together, they roost together, and they visit nests together. And of course, the question is if they are genetically monogamous or not. But we expect to have data on that in the near future. But this is something interesting, because one possibility is cooperation of males with females at the time to prospecting or accessing host nests. Now we move to defenses and counter-defenses during the nestling stage. And just to give some background information, the way in which brood parasites trick parents differ. But we can say that you have 4 non-mutually exclusive mechanisms. One is begging exaggeration. The other is to tune the begging calls to the parent offering communication system. This requires some learning of parasite chicks. Other possibility is sensory exploitation. They use signals that are not part of the host parent offering communication system, but exploit some kind of sensory bias that the host has. And finally, you have host chick mimicry, in which the parasite chicks have visual or vocal similarity with host chicks. And that similarity evolved to evade host discrimination against chicks which are non-mimetic. So we knew that, as I showed you before, that the plumage of the parasite chick is similar to that of host chicks, and that the vocal begging calls are also similar. So our question was if the parasite chick was as good as the host chick in stimulating the provisioning of food by the parents. So we did an experiment in which we broadcasted begging calls. We put a loudspeaker inside the nest, and each nest had 4 baywing chicks of 6-7 days of age. And every time one parent went to the nest with food, we broadcasted begging calls of an extra baywing, 1 screaming cowbird, 1 shiny cowbird, or 1 control. The control was without playback. So we did the playback of 1 extra host chicks, 1 specialist brood parasite chick, 1 generalist brood parasite chick on nothing. And we did this sequentially. So each nest received the 4 treatments. And again, the order of presentation was rotated. And the variable we recorded was the frequency of feeding visits. And we collected this data, putting a micro camera inside the nest. And the results show that the number of feeding visits with the control, that is without playback, and with the shiny cowbird playback was similar, while the number of feeding visits when we put the playback of 1 baywing and 1 screaming was higher. So in some way, screaming and baywing playback elicit more visits from the parents, and parents react in the same way to playbacks of the specialist parasite and the host. However, screaming cowbirds and shiny cowbirds do not differ in growth rates. So shiny cowbirds, although they are not good stimulating parental provisioning, growth normally, they are growth curves of males, the black dots, and females, the white dots, and the other line is the host, and they don't have problems obtaining food while they are inside the nest. So although begging calls are more effective, begging calls of screaming cowbirds are more effective than those of shiny cowbirds manage to get enough food, probably because they are using the stimulus of host or the specialist parasite begging calls. And the question was, this begging calls is innate or is learned in some way. So we did a translocation experiment in which we translocate screaming cowbird chicks to nests of a species that they don't parasitize, the mockingbird, the chalk-brown mockingbird. And you can see the begging call, the spectrograms of the begging calls of screaming cowbirds in baywing nests when they are 4 or 5 days of age, and when they are 8, 10 days of age. The begging calls of the screaming cowbirds in the nest of mockingbirds, again, and this is the begging calls of baywing, and this is the begging calls of mockingbird. And as you can see, there are some small differences in the structure of screaming cowbirds raised by baywings and raised by mockingbirds, but they are quite similar. So to test if these begging calls are a good stimulus for parents, we did an experiment in which we broadcasted from a speaker close to a nest which had baywing nestlings, one begging call of a screaming cowbird raised in baywing nest, one begging call of a screaming cowbird raised in mockingbird nest, and begging calls of baywings, and there were no differences. So the screaming cowbird begging calls, the screaming cowbirds raised in mockingbird nest, were as effective as those of screaming cowbirds raised in baywing nests. So we can conclude that there are good evidences that screaming cowbirds begging call is in some way innate, that is not learned during the time they spend in the nest with baywing chicks. So in the last part of this talk, I'm going to talk about defenses and counter-defenses during the fledgling stage. And this is when the most interesting thing happens, because shiny cowbirds chick survive without problem while they are inside the baywing nest. But once they fledge, baywing stop providing parental care. So we did a, we try to follow, and this is not easy, follow chicks, follow young when they fledge. They are quite elusive, and it's not easy to detect them, but we follow during the first week after fledging chicks of baywings, screaming cowbirds raised by baywings, and shiny cowbirds raised by baywings, and all except one shiny cowbird disappear. And the only shiny cowbird that survived, and we assume that disappear means that they died, the only case of shiny cowbird that survived was because it was fed by other species. They start, begging to another species and manage to obtain food and survive. So apparently at the time, chicks fledge from the nest, the vocal and visual cues become more important, and baywing parents discriminate between their own chicks and non-mimetic chicks. And this is just to show you in more detail the development of the plumage in the host and in the parasite. This is an adult host, this is an adult parasite. This is about 14 days of age host and parasite. And the only difference you have when they are young is that the parasite have these black underwing coverts, and the host have his black lores and black around the eye. But it's almost impossible to distinguish host and parasites. And as I mentioned before, at the time they stop receiving parental care from foster parents, they start molting, and they are kind of harlequin, and after 4 or 5 months, they get the adult plumage, which is quite different from the plumage of the juvenile. So one question is that if this that looks mimetic, similar to our perceptual system, is similar for the perceptual system of birds, and as probably most of you know, birds have tetrachromatic vision. They have 4 different types of cones, and they have cones that detect light in the near ultraviolet. So the way to measure similarity in plumage color is using reflectance spectrophotometry. And we did that in 8 different parts of the body. And then we use a complex procedure to analyze the data, which is called the Boroyev-Osorio discrimination models, that take into account the kind of pigments that have cones and the way in which the brain processes the visual information. And at the end, what you get is that everything which is below this dotted line is differences that are not detected by the perceptual system of the host. And here we compare baywing versus baywing, so 2 chicks of baywing, baywing versus screaming, baywing versus shiny, and screaming versus shiny. And you can see that the only differences that the perceptual system of the host can detect are differences between baywing and shiny, or screaming and shiny. So for the host, the plumage coloration of baywings and the plumage coloration are of screaming cowbirds are identical. They can't discriminate between both chicks or young. And as regard the vocal information, I just give you, this is the begging call of a baywing. This is the begging call of a screaming cowbird, quite similar. And this is the begging call of a shiny cowbird. You don't need statistics to say that they are different. Just look in the spectrograms, they look quite different. But if you make a discriminant function analysis, you get a clear separation between the shiny cowbird begging calls and the screaming and baywing. And even there are small differences between screaming and baywing calls. The peak frequency is higher in screaming cowbird chicks than in baywing chicks, but begging calls are very, very similar. So it looks like that we have both visual mimicry and vocal mimicry. But of course, the question is this vocal signal good enough to attract baywing parents? So we did, again, one experiment in which we broadcasted calls of baywing, screaming cowbird, and shiny cowbirds from a loudspeaker close to the nest, in nests which have chicks at the time they fledge. And again, was a within nest design. Each nest received the 3 treatments 3 minutes of baywing calls, then 10 minutes of interval, then another 3 minutes interval, another 3 minutes. And we repeat this in 15 nests. And the variables we measure were the latency to approach the speaker, the number of baywings recruited. As I mentioned before, they are cooperative breeders, so some nests have 1 or 2 helpers, the time spent close to the speaker, and the number of interactions with the speaker. And the results show clear differences, not only between baywing and screaming with shiny, but also between baywing and screaming. Screaming begging call were more effective attracting parents than the own begging calls, the begging calls of their own species. This is lower latency in screaming than in baywing, and of course, lower latency in baywing than in shiny. More adults recruited by screaming than for baywings, and baywings than shiny. More time close to the speaker, and more interactions with the speaker. These are not independent variables, but the 4 variables have the same pattern. And the question is that why screaming cowbird begging calls are more effective than baywing calls? And in some way, the results suggest that there is some kind of host manipulation through the structure of the begging call, and perhaps this higher peak frequency is something which is very attractive for parents. To summarize, sorry, the last slide of this part is one experiment, what we did, expecting quite different results. Our rationale was that what happened if a shiny cowbird is alone in a nest of baywing? And what we expected was that they will die because the begging call is not effective at stimulating host parents. So we compare the survival and the feeding rate that receive shiny cowbirds that were reared alone in baywing nests and with baywing chicks. And we have no differences at all. They receive food at the same rate when they were alone that they were when they were sharing the nest with host chicks. And they grow similarly when they were alone that when they were with host chicks. And which is more strange, after they leave the nest, they receive parental care from parents. And this suggests that there is kind of context-dependent host discrimination abilities. The host responses towards non-mimetic fledglings in some way depends on whether they have fledged with or without host chicks. So as a summary of this part, I try to show you different defenses and counter-defenses at the 3 nest stages: before and during the egg stage, during the nestling stage, and during the fledgling stage. And although we can discuss if some parasites' behaviors are counter-defenses or are just adaptations, general adaptations to brood parasitism, it seems that vocal mimicry of nestlings and visual and vocal mimicry of fledglings are counter-defenses of the parasites to defenses of the host that are the discrimination between non-mimetic begging calls on non-mimetic plumage coloration of fledglings. So this is most of the talk I wanted to present, but I mentioned at the beginning that baywings were the main host of screaming cowbirds. And when I say the main host, is that in most distribution of screaming, they parasitize this host, but they can use alternative hosts. And this is the distribution of screaming cowbirds. And you can see these arrows indicate that this is expanding to the north of Brazil, but it's present in an area where there is no host. And they are using this alternative host, the choppy blackbird, for moving towards the north of Brazil. And they use another alternative host, which is this brown and yellow mashbirds. And we study the interactions between screaming cowbirds and choppy blackbirds in the northeast of Argentina, in Formosa, the same place where Eduardo works. And it's obvious that the adaptations that allow screaming cowbirds to parasitize baywings are not useful when they are using this alternative host. So this plumage coloration is mimetic with baywing chicks, but not with choppy blackbird chicks. And this is young, start to mold the plumage, have two small patches of black in the chest with the choppy parents. And what we could show is that it uses regularly, this is the timing of breeding for choppy blackbird. They use choppy blackbirds from early October to the end of the breeding season. This is proportion of nests along the breeding season, and the stripped part of the bars indicate frequency of parasitism. And this is the timing of breeding for baywings. So in the same place, they are using the two hosts. And one study we did with mitochondrial DNA, which is maternal inheritance, showed that the network of haplotypes differ between screaming cowbirds raised by choppy blackbirds and baywings, which is an indirect evidence of host-specific races. So apparently host use is not random. Some screaming cowbird females parasitize choppy blackbird and others parasitize baywings. And this is a long-term study, more than 10 years of data. This is the frequency of parasitism. You can see that the frequency of parasitism of choppy is high. Some years is relatively low, but some years is up to 70% of the nest. And again, the intensity of parasitism is quite high, up to 4 or 5 eggs per nest. Here you have 3 eggs of choppy and 3 eggs of screaming cowbird. And the success in choppy blackbirds is very good. This is the number of screaming cowbird eggs laid during pre-laying. And if you remember, I mentioned that one adaptation of baywings was that unpredictable start of egg laying, and synchronization is much better. Synchronization of the screaming cowbirds with host egg laying is better in choppy blackbirds than in baywings. And although the hatching success, the proportion of eggs that hatch is relatively low, the survival of eggs and the survival of chick is quite high. So if you consider all the variables and you just calculate what is the proportion of chicks or young per egg laid, well, the result is as good in choppy blackbird or even better in choppy blackbird than in baywings. So choppy blackbird is a good quality host of screaming cowbirds. And the question is, why? Which are the characteristics of this alternative host? And this is one of the questions I like to emphasize. We did in the past some experiments in which we translocated screaming cowbird chicks to host of the shiny cowbird. And we did it with Chuck Brown mockingbirds and with house wrens, 2 very common host of shiny cowbirds. And in both cases, the screaming cowbird chicks didn't survive. In the case of Chuck-Browett mockingbird, which is a larger host, because competition for food was intense and they died of starvation. And in the case of house wrens, because they were parasitized with Philornis, which is a fly, that it's an ectoparasite that produces a high mortality. But this alternative host that I mentioned, the brown and yellow marshbird and the choppy blackbirds, as well as the baywing, the three have helpers at the nest. And another two alternative hosts, which are not, they have not studied yet, the Austral Blackbird in Chile and the yellow rump blackbird in Brazil, they are parasitized by screaming cowbirds, and they also have helpers at the nest. And not all the icterids, all these species are the same family of birds, but are the only species in the family which have helpers at the nest. So apparently, competition for food with host chicks perhaps is a selective force that prevents the successful use by screaming cowbirds, the successful use of other hosts by screaming cowbirds. And finally, I'd like to show you what to me is a nice natural experiment as a result of habitat transformation. This is Brazil, and these red dots are places in which Brazilians took photographs of screaming cowbirds and pale baywing. There is a very nice place which is called Wikiaves, which is a citizen science project in which bird photographers upload the photographs. There are very nice photographs of all birds of Brazil. And because these photographs are georeferenciated, you have the location in which the photo was taken, and you can just monitor the range with, of course, some restrictions, but you can detect the range of one species. And this is the places where you got photograph of screaming cowbird. And I mentioned at the beginning that the baywing distribution was split in two areas. And at the beginning, people thought that they were the same species, but now they think they are different species. This is the pale baywing, Agelioides fringillarius. And as a result of this movement of screaming cowbirds towards the northeast of Brazil, this species now is in contact with screaming cowbird. This species also have helpers at the nest, and of course, it is parasitized by a screaming cowbird. And this recent sympathy provides a nice natural experiment to study host defenses and eventually parasite counter defenses in a new host parasite system. So my final remarks are that antagonistic interactions between brood parasites and their host are a very good model to study coevolution. And over the past, I would say between 30 and 40 years, we obtain a great amount of knowledge about the reciprocal adaptations, which are driven by brood parasitism, and systems like the one I show you, in which the parasite at host specialists are very suitable to study the coevolutionary relationships and how these interactions resulted in defenses and counter defenses, resistance or tolerance to parasitism. And as I mentioned, the use of new hosts by brood parasites, and normally it is as a result of habitat transformation that parasites expand their range, provides a unique opportunity to study the evolution of host defenses. And I just like to acknowledge the co-authors of these results, Maria Cecilia Di Marsico is working with baywings since the beginning of this century, and Romina did most of the radio tracking studies. Cynthia did most of the work, molecular work. Juan is now studying the cooperative breeding system in baywings. Alejandro did the work in Formosa. Florencia did some experiments. And bueno, Rosalind Glow was an Australian PhD student that worked with us for several years under the supervision of Alex Kasenik. And that's all. And thank you very much. And I'm open to answer any question.
Thank you very much, Juan Carlos. Thanks for a wonderful presentation. Very quickly, just a reminder, if you want to stop sharing your screen, Juan Carlos, then I can.
Yes,
we can
let me find.
Very quick reminder, I invite you to type in the chatting window a question mark that will tell me that you have a question for Juan Carlos. And once again, our colleagues joining us through YouTube, please, we'd like to hear from you as well. And our students and young colleagues joining us either through YouTube or here in Zoom, please, we definitely want to hear from you. So with that, I thank again Juan Carlos for a wonderful presentation and look forward to the questions we may have. And the first one comes from Karsten. Karsten, please.
Yes, hello, I'm Carsten Schradin from the CNRS in Strasbourg, France. Thanks for interesting, fascinating talk with lots of information. I have one question, maybe you answered it and I missed it, but I don't think you discussed it. One of the counter strategies of the host is, like you showed very nicely, the unpredictability in egg laying.
Mm-hmm.
Do you have any idea what costs this induces on the host? I mean, it must be kind of costly that they do it or not.
No. Yeah, of course, there is one cost, that is that your chicks have less time to until they reach the autumn, but apparently I wouldn't say it's a big cost. In fact, perhaps, and that could be one possibility, that this unpredictability is more frequent in early nesters than in late nesters. We didn't analyze those data. Perhaps now we have enough data to test that hypothesis, that they are more, the pre-laying period is longer if they are at the beginning of the breeding season than if they are breeding late in the season, where the cost probably could be higher.
Thank you.
Thank you, Karsten. The next question is from Bragg. Please remember to, if you could be so kind, of reminding the participants where you're visiting us from, I mean, your affiliation and where you are. That helps us build a community of fine participants.
Yeah, I am Raghavendra Gadakkar from the Indian Institute of Science in Bangalore. Thank you very much. I learned a lot from this talk. I know a little bit about the cuckoos, but didn't know anything about cowbirds and baywings, so I learned a great deal. I have two questions. The first is, you said that if there are too many parasite eggs, then the host will simply abandon the nest and start all over again. But you said when she starts all over again, the next time the rate of parasitism is somewhat less. And I was wondering why it becomes less in the second attempt compared to the first attempt. Does it have anything to do with the season? That was my first question. My second question is, it seems to me that this screaming cowbird and grayish baywing seem to be so well adapted to each other. And I'm wondering, is there any chance at all that the host benefits in some way by having the parasite eggs, some parasite eggs and chicks in its nest? So for example, can we compare the survival and growth rate of baywing chicks in unparasitized nest versus parasitized nest? Is there any hint of some indirect advantage.
Okay, as regard the first question, why intensity of parasitism is lower in the replacement nest? Well, if you assume that the number of eggs have a kind of normal distribution, what they are ejecting are the clutches which are in the right side of the normal distribution. So in some way, what you expect is a lower intensity just by chance. It could be that in the second nesting attempts, they also receive a large number of eggs, and again, but it's quite unusual. So it's just they don't know because they don't recognize parasitic eggs, but detect that this is an abnormal clutch size and reject the whole clutch. The second question was
whether there's any possible advantage of
benefits of being parasitized. Well, this is the title of a famous article in Nature by Neil Smith, which was highly questioned in the past, in which he suggested that the giant cowbirds remove botflies from host chicks, and that was the benefit. I can show you then, if you are interested, videos of baywings removing botflies from host and parasite chicks, but there are no evidence at all that there are benefits of brood parasitism in any brood parasite host system studied until now. In our case, the costs are not very high. Main cost, as I mentioned, is the destruction of eggs at the time screaming cowbirds parasitize the nest, and this is another story, but cowbird eggs have thicker egg shells, probably as a result of this interspecific competition. It's a different story, but this is the main cost that hosts pay. And one interesting question is why you have antiparasitic, the more effective antiparasitic defense is at the fledgling state. And at that time, the host paid all the cost of parasitism. It's not avoiding any cost. So it is strange because what you expect is that defenses reduce the cost. But in this case, the cost is only provide food to non-mimetic fledglings in a species in which you have cooperative breeding. So the cost for the parents is lower because the parental care is shared with non-breeders. So it's strange, but on the other hand, they discriminate non-mimetic chicks. They do not provide food to non-mimetic chicks. So we don't have a good explanation for this.
Thank you.
So just to really get our students asking questions, we really want to meet you, want to hear from you. I'm going to ask Suleima to bear with me, and we'll just first take Jin-Yu Q, who's an active participant in the seminar. Jin-Yu, please, your question.
Hello, I'm a new PhD student of Carsten based in Strasbourg, France. So my question is, have you ever observed any evidence that this specific parasiting of the cowbird may reduce their successful rate when they're parasiting other species, like you said, or they are parasiting new hosts?
If they reduce the success of, sorry?
Their success of when they're parasiting other species.
Yes. The impact is the same, is lower because the two species that have been studied are the brown-headed cowbird, the brown and yellow marshbirds and the choppy blackbird. Both are larger. They are about 70-80 grams versus 45 grams of screaming cowbirds. So the main cost is egg extraction, but it's relatively low cost. And these two hosts have no antiparasitic defenses at all. The other two species that I mentioned, one is the Austral Blackbird, and that is another interesting case because that happened in Chile, where you don't have baywings, and screaming cowbird have been introduced probably as a result of illegal trapping and pet trade, and it's expanding in Chile using this new host that again have a cooperative breeder. So perhaps, as I mentioned, competition for food is one, because at the end the question is, which are the advantage of being an specialist brood parasite? If you compare shiny cowbirds against screaming cowbirds, shiny cowbirds is a highly successful species. It's invading United States. It expands throughout Caribbean. Is really in every new transformed area in South America. You have shiny cowbirds, and it's incorporating new hosts to the list of hosts. So if you are a specialist, you depend on one, your population depends on the population of one host species. If you are a generalist, you can switch host, and it's a better strategy. So why screaming cowbirds become specialists? This is one of the open questions.
Okay, thank you.
Thank you. Suleima?
You can go on to Ricky and come back to me if you want
Go.
to, Eduardo.
Now we have time. Go ahead.
Okay. Okay. Suleima Tang Martinez, retired from the University of Missouri in St. Louis. And thank you, Juan Carlos. It was really an interesting talk. I have two questions. The first one, I was really intrigued by the fact that so many of the, or maybe all, I wasn't sure, of the hosts for the screaming cowbird have helpers. And I was wondering what role the helpers are playing. Obviously, if you have helpers, it means that the parasite may be getting more food, the chicks. But I wonder if the helpers play any role in defending the nest against the screaming cowbirds. So is that relationship of having helpers more complex than, you know, simply feeding, there being more individuals to feed the nestlings? And then the second one goes right back to what you just said, and you said it was an open question as to why you have these extreme specialists versus generalist parasites that, you know, are extremely successful. And you said it was an open question, and I'm just wondering if you can expand on that, and if you have any thoughts as to, even preliminary thoughts as to what might be going on in terms of the evolution of extreme specialists versus more generalist brood parasites.
Okay.
Thank you.
The first question, there is a paper a few years ago by Australian group in Science, I think it was, relating cooperative breeding and brood parasitism, and show an association. But that association came from mainly studies in Australia and in Africa. And in Australia is particular in the sense that most cooperative breeders are from Australia. So the argument is that parasites prefer cooperative breeders because cooperative breeders in some way guarantee more food for chicks, and cooperative breeding could be a way to avoid parasitism if helpers are present at the time egg laying occurs or parasitism occurs. And this is not the case in several species of cooperative breeders, in which helpers join the breeding pair late during incubation or after hatching. As regards the second question, I just can speculate. I can imagine that perhaps in the past there were competition between shiny and screaming cowbird for hosts, and in some way the baywings was kind of refuge for screaming cowbirds, and this result in a coevolutionary process and the specialization of screaming cowbirds in baywing. But we don't know how recent is the use of new hosts. As a color note, Francisco de Azara was a Spanish in the 18th or 17th century who was one of the first people that describe birds. And if you read the description of the chick of the choppy blackbirds, it is a description of a screaming cowbird. The description of the plumage coloration of the choppy blackbird is the description of the screaming cowbird. So in 1800 something, choppy blackbirds were parasitized by screaming cowbirds. We don't know when they started to parasitize brown or yellow marsh birds, or probably Austral blackbirds was recently. Sure, Agelaius fringillarius, the pale baywing, is in the last 10 or 20 years, but we don't know what happened 500 years ago, before the arrival of Spanish people to South America, if they were using only baywings or they had at a low frequency these alternative hosts. So if I had to bet, I would say that perhaps it was the result of interspecific competition with shiny cowbirds, and because screaming are less competitive, and perhaps we can do experiments to test that, they ended in baywings as a refuge.
Gracias. And it seems that it's almost like, the real specialists are literally putting all their eggs in one basket. And if anything goes wrong, the whole species, the parasite could also be in trouble.
Yeah.
Okay, thanks.
Thank you.
Thank you, Suleima. The next question comes from Ricky Laser. Ricky?
Hey, can you hear me?
We can hear you well. If you can introduce yourself.
Awesome. My name is Ricky Laser. I am a second year now in Alexander Ophir's lab at Cornell. And I have two questions for you, if that's all right. One of them is if the adult calls of cowbirds, or of the screaming cowbirds, sound like baywings as well, or if there are differences there, and when, and if you know anything about how these develop. And the second one is about nest temperatures. That's my own bias for asking, so it's totally okay if you don't know, because that's something I'm looking into a lot right now. Specifically, whether the screaming cowbirds, part of the reason maybe that they are specialists is perhaps their incubation temperature or some such thing is very specific, whereas the more generalist birds maybe are okay at like more variable nest temperatures.
Well, I start with the second question and then I ask you for the first one because I don't remember that. But no, I don't think, well, we never measured temperatures in nest of hosts. Baywings don't build nests, they usurp nests of other species. Most nests are from furnarid species, but you have a huge variety of nests. So what I expect is that because these differences in nest, you don't expect, it's not like an ovenbird which have a particular nest, but we never measured temperatures. So could be, I don't know. And the first question was?
Yeah.
Begging call, something about
adult, the adult baywing and adult cowbird calls, and if they are different or similar.
Yeah,
or...
quite different as the plumage of the adults. The adult calls of baywings, it's they are totally different. So it's like the plumage. You have a similarity until they emancipate from the foster parents, and at that time they molt in the adult plumage, and they start with the adult call. They are quite different species. And at the beginning, the baywing was included within the genus Molotrus, and they thought it was a kind of species within the same genus, but once taxonomists start to doing molecular phylogenetics, it became obvious that they were quite different species. Now it's a different genus, and a bit far from the group of Molotrus. So it's interesting, all the process of gene regulation that result in plumage similarity, and how this changed at the time of emancipation of foster parents.
Thank you.
Thank you very much.
Our colleagues joining us through YouTube, please don't hesitate to let us know if I don't know if anybody's checking where we have some questions from them. In the meantime, the next question comes from Karsten.
Yes, I have a question for the social monogamy in screaming cowboys you mentioned twice. And just you might have said it, but just for me to summarize it again, I have two questions for this. First, what evidence do you have that they do live in pairs, that they are forming pairs? And second, you said male-female cooperation during the egg-laying phase could be important. Is this a hypothesis, or do you have any evidence in this or any other species how this cooperation might look like?
No, the second question, we don't have any evidence. It's just a hypothesis. As regard the social monogamy, we have good evidence, or relatively good evidence. They remain, when you trap, we use normally walk-in traps, and normally female are trapped and the male remain around, or when you use mist nets and one individual is trapped in the mist net, the other is around. Normally we trap one male and one female. And at the beginning, there was some kind of circularity, because we trap one male with one female and then we follow them, and they were associated. So we did this kind of social network analysis, in which we radio tag 10 pairs of males and females, and of course they were trapped together, but then we follow all possible associations between these 10 males and 10 females, and the associations were between the male and the female that were trapped together, and they had the same home range. And so we are quite convinced. The problem we have, and just a short description, the way in which you did this have some problems, caveats, because we are receiving in 4 different receivers the signal of the tag. And we did a kind of signal map, and we did manually with radio tag at 4 meters of altitude, moving along the grid, and we got 600 different points, and then we use a kind of, we use artificial intelligence to estimate the position of one bird from the 4 signals we detect simultaneously in the receivers. And that has an error of a couple of 100 meters. So we don't know that if they are exactly one together, if they are at 2 meters of distance, of 10 meters or 100 meters, we know that are within 200 meters of distance. We did this manually, tracking pairs of tracking one individual, and in that case, we detect both male and female together, but this is time consuming. And the other line of evidence is what we put this omnidirectional antenna in baywing nests and had males and females radio tagged, and they visit the nest together. But it could be also that there is, mate guarding from the male. The male is just following the female during the whole breeding season. At night, they stay together in the same place in the roost inside. And the roost inside is about 600 meters long. In 90% of the nights, more than 90% of the nights, they are close. We don't know if close is, 1 meter, 5 meters, 10 meters, but they are close. We are just with antenna trying to detect the maximum signal of a bird, which is 10 or 15 or 20 meters in the roost inside. But we don't know what happened at the genetic level.
Okay, thank you.
I go in bed for genetic monogamy.
Thank you. The next question comes from Michael Taborski. Michael?
Yes, Michael Taborski from University of Bern in Switzerland. If your parasitic species is so highly specialized, of course, regarding the host species, of course, there needs to be a mechanism how they recognize and how they find their hosts. It's a bit similar to the gentes in European cuckoos that need to parasitize the right kind of host. Do you have any idea about how they find their hosts?
Well, as you probably know, most evidence is indirect. The only case in which we have direct evidences of an imprinting, a filial imprinting, is with Vidua, the pain studies. What we have is that there are genetic differences between screaming cowbirds raised in choppy blackbird nest and screaming cowbird raised in baywing nest, which is an indirect evidence of host-specific races. What I assume, but I have no evidence, is that kind of imprinting process like the one in Vidua is what happened in most parasitic birds. They have an imprinting with the foster parents and use that as a search image as adults. And this host imprinting works better in specialist brood parasites than in generalist brood parasites. Generalist brood parasites have apparently host imprinting, but they can include new hosts in their host list, as shiny cowbirds and brown-headed cowbirds have shown. In this case, it's interesting. It's just anecdotal, but baywings sometimes use nest boxes. We offer them nest boxes, but they prefer the nest of Furnariid species, which are much more complicated. But sometimes they use nest boxes, and sometimes they fight for the nest boxes with house friends, for instance. And screaming, once the baywing occupy the nest box, are close to the nest box all the time. Before, they just avoid that nest boxes. So they recognize baywings as something attractive and look for baywings. And in that case, it's not habitat imprinting or nest characteristics imprinting, because nests of baywings are quite dissimilar. So if I had to bet for something, I would say that imprinting in foster parents is the mechanism the parasite used to search for hosts when they are adults.
Okay, yeah, thank you. That's very interesting, because in European cuckoos, I think the evidence is stronger for habitat imprinting than for host
I know.
imprinting.
I know.
Great, thank you.
I am next with a question, unless there's someone else who to have any student. Please type a question mark if you want. If not, in the meantime, Juan Carlos, you briefly touched on the use of these tags, passive data loggers, and using artificial intelligence. And this is something you and I discussed now some time ago when we were trying to implement similar technology in Formosa in the forest to try to learn something about the solitary floaters. Can you share a little bit more how those things, those attempts at implementing some kind of AI approach to the data you collected have gone? And if there's, I'm curious about the specifics and the details. I appreciate how one of the very first things you said is that they come with errors, right? And you just mentioned a couple 100 meters. And sometimes we get maybe too excited about technology without paying enough attention to the uncertainty that there is associated with these devices. So can you share some more of the specifics, since we have our own trouble with those?
Not very much on artificial intelligence. We did this work with people of the computer department, but I can send you the manuscript, which is now in revision in one journal. The main problem is to have a good calibration. The transmission of tags depends on vegetation, altitude. There are a lot of noise. The problem we have is that you can put one data logger with the GPS to a cowbird, because the smaller one is 6 grams, and that is 15% of the body mass of a cowbird. But if you work with monkeys, you can put in a monkey everything. So in that case, my suggestion, of course, is much more expensive, is to put some device with GPS and Bluetooth and just collect the data of the position with an error of a few meters. If you need to use this system of automatic detection, it is important the calibration curve, and I think the use of drones could be a very helpful tool, because you can get very many points. So you put an radio transmitter in a drone and collect the signal in as many positions as you want, as many altitudes as you want. And
you mean a receiver in the drone, not a transmitter? But
no. You have 4 receivers. You have a grid of receivers, and you put a radio tag in the drone, and the tag is moving like a bird or like a monkey.
Okay, for calibration. Got you.
And in the calibration, you have, instead of doing manually, in which you have to work with a pole and a tag, and this is terrible. You can get hundreds of thousands of points, and this improve what the people in the computer department told us is that, oh, what a pity, you have only 600 points. And to get 600 points was terrible. But now with the drone you can get thousands of points, and this apparently improved very much the quality of the data. But if you have the possibility to put a GPS in a monkey, it's much better.
Thank you. We have a question from YouTube. Lauren?
Yes. So I have a question from Claudio, who is at the Universidad Latina in Mexico City. Claudio writes, thanks for this nice talk. The question is the inability for recognizing parasitic eggs by baywings, would that be a result of the eggs of a host and parasites being almost identical to the human eye at least, or because keeping in memory how many eggs an individual has laid is enough to know it has been parasitized or not?
Well, counting eggs apparently is not a strategy to detect parasitic eggs. There are no evidence. The only evidence I think of counting that I know in brood parasitism is in black-headed, in a host or in coots. It's a paper of Bruce Lyon in Nature in the 90s. They recognize eggs using the color of the eggshell. But one of the problems is that they are cavity nesters. So the light conditions inside the nest are not good to detect differences. We can recognize, and if we can recognize, for sure birds could recognize, but you have some host species which are very good detecting eggs which are different, and you have egg signatures to recognize their own eggs, and some host species that don't discriminate between eggs which are quite different. In this case, I don't know if there were no selective pressure to discriminate eggs, or there is not such possibility because the light conditions of the nest preclude this kind of this is not a reliable cue in the nest in the light conditions of most baywing nests.
I don't see other questions. I just let everyone know, or I'm getting started with a huge storm, and it's not unusual that I lose power, but both Lauren and Karsten, you guys are co-hosts. Should that happen? Do we have any other questions from the audience? If not, we could probably, Juan Carlos has been meeting with some of us from very early morning today. We thank you for that. It's been fantastic. So if there are no more questions, what I'm going to do is I suggest, Lauren, Karsten, that